As we are first time here in Pages conference, I'd like to quickly introduce our company. We are located in Regensburg. The university is much younger than the one in Oxford, but we do have a stone bridge that's almost as old. And if you turn to the right and go about 25 kilometers, you're directly at our doorstep. So what we do, as I already said, we work with mass spectrometry. We produce products as standards for mass spectrometry, but we also offer services. And we really focus on absolute protein quantification. So not only comparing more or less into samples, but really putting a specific concentration on each protein. And we had this nice slide from Martha this morning. She allowed me to show it again. So here it is. So when we heard a lot about the pages, and this is the clinical phage cycle more. So we are more working on the part regulation. There you at some point you will have to do impurity testing, or if you want to go to a certified clinical phage, and you will have to repeat it during the trial, because every time you produce a new batch of your phage, you will have to prove that it's as good as the last batch. But what we would like to recommend is to start already there at the production. So when you go into making up your production process and purification process, maybe it's a good idea to already look there at your impurities, so you don't have to go back later on. So what are these host cell proteins? We heard already of LPS, that's another problem, but there's also more that is released from your host cells, from your bacteria where you produce your phages. And these, so host cell proteins in general, are non-product related proteins. These are remnants of your host organism for production, and they co-purify with the therapeutic product and are typically present at low levels, at least for purified proteins. Phages looks a bit different. I will show you. So why are these a problem? They can negatively affect the stability of your product. Maybe protein input can degrade your phages or can render them inactive, and also they can reduce the stability, storage stability of your drug. But they can also directly cause adverse effects in patients. Could be immunogenic or allergic reactions, or they can even be directly toxic or infectious, like prophages in your case. So there is some regulatory demands. So you will, when you go to the clinics, you will sooner or later have to do some impurity testing. This is still a developing field, so there's not so much. From the EMEA, there's a concept paper, and there will be a guideline someday, but I don't know when. But there's a new chapter in the European Pharmacopoeia where they also define that you have to test for endo and exotoxins, for host cell proteins, host cell DNA, and temperate phages in phage preparations. Chapter 5.1, and the US Pharmacopoeia. This is a general chapter about impurity testing. There they recommend really measurements by LC-MS and using internal standards. They also recommend to test early during production stages, so in process development. So in the case, we measure a lot of purified proteins, but for phages we have some specific challenges. So for one, you lyse your cells. So you get a lot of, you don't have only a cell supernatant, but you really lyse your cells. Everything in the cells will be released into your solution and will be, so you start with really high levels of contaminants at the first place. Then therapeutic phages may have to produce a pathogenic host that may produce toxins. So you need to control that we successfully remove these toxins. Then there are prophages. Prophages might be reactivated during phage production, and you also don't want to have these in your final product. And then there's the problems of hitchhikers, which also apply to recombinant proteins. For the phages, there's just more space where they can attach to. So hitchhikers are called recombinant host proteins which attach to your drug product, to your phage. For phages, there might even be incorporated into the phage particle in some place. So what we like is to measure all these contaminants by mass spectrometry. Of course, you can develop single ELISAs for all of these targets, but you can just go to the mass spectrometer and you can measure a lot of these in one single experiment. Typically, this is done on peptide basis. You degrade your proteins down to peptides and you separate them and analyze all of them simultaneously in the mass spectrometer. For recombinant proteins, it may look like this. You get 95% or so your main target protein, the drug product, and then you have some minor contaminations from your host, in this case, E. coli. For phages, it looks a bit different. So you have not one phage protein occurs, you have a lot of different phage proteins and phage, and you have a lot of HTPs in there. So this is from some test data we generated in our lab. And then we have a special technique so we can not only identify all these contaminants from your host organism, but we can also quantify them and really put a concentration. I'll show you how that works. So we generated some test data. I can't show you the data from customers. So we, for the first time, propagated some phages, T4 and T7, both in the same host cell, CoV-DSM 613. And we
Okay. Yeah, the nice thing is we can directly add it to a biological sample, digest it together with the sample, and measure it together. These are labeled by stable isotopes, so we can rule out which is the standard and which is the native protein. And we can put up to 50 peptides on one of these constructs. What we did we incorporated into the yeah. Okay. So we integrated 2 or 3 peptides for each of these targets, and we labeled it. And now we have these 2 constructs with about with 100 peptides covering 48 proteins. And what we do is now we get 1 signal from the standard and 1 signal for the native protein, and this is from 1 measurement, and then we can quantify because we know the abundance of the standard, and we can quantify the other proteins. These are now, this I showed before, and these are the proteins in our purified product, which are covered by the different Q concats. So we can, to this, we can now directly put a concentration. So here we have the highest more than 400 nanomolar concentration. And we could even take all the others and repeat the question. Yeah. What was the next step after that? Then we produce it in E. coli, actually. So we produce it as a recombinant protein. We purify it. It's produced with isotope labels. So we incorporate. Then you can add it to your samples, right? Oh.
Thanks a lot for a very nice talk. My question is, I understand the point that it becomes more and more essential to have a pure product But how scalable is this assay, and also how how diversified can it be? Like apart from E. coli, can it be expandable to other species?
Well, yeah, it can be easily adapted. Basically, what we need is a good genomic database. This should be present or should be made up before we can start. But we don't even need the standard absolutely to look at your proteins. We can develop then on the go. So typically, we start out with an untargeted measurement where we can just directly run as soon as we have the genomic data present. Then later on, we can develop a customized, like QConcat for your species. And we should, of course, also include like toxins, for example, can be directly included. We don't have this on this off-the-shelf product, but this can, of course, be done.
So higher volume of product?
Yes, yeah. So we don't need much of the product. We need about, what we measure on the machine is 1 microgram about the product. All we need. But it doesn't matter how large your batch is, basically. So.
Yeah, thank you so much for the talk. The concatemer solution is really clever. And I think immediately applicable to all of us. No, but I guess I have a more phage-oriented question. The DSMZ613, that's just Lur's E. coli, which has 2 quite active prophage in it.
Okay.
Now, both of which should be excluded by T4, but one of which definitely isn't excluded by T7.
Yeah.
So there should be at least 1 prophage in that data.
Okay.
No, but around what would you expect the limit of quantification to be for the, like, at least as measured in PFU for a temperate phage element?
Yeah. So I have to be honest, these are like really quickly made example data. So it didn't look so much into detail. And it was a screening. So this was just a kind of mass spec experiment, a DIA method, where we do get quite a good resolution. But it will improve if we have these standards made and we target really the machine on the proteins we want to see. The detection level will be better. So we typically get down to about 1 nanomolar concentration. We can go lower when we go targeted. So maybe going down to 0.01.
The PFU estimations then depend on the copy numbers.
Yes, so you could do PFU estimations from that, but as we heard before, qPCR, it runs into the same problems as qPCR. You wouldn't be able to discriminate active from inactive phages, right? Yeah.
Okay, thank you again.